Donc bonjour à tous. Donc aujourd'hui, nous recevons Anaïs Baudot qui va nous présenter ses travaux. Sorry, I switched to English now. So Anaïs Baudot obtained a PhD in bioinformatics in 2007 from the Aix-Marseille University, where she worked on predicting protein function from protein-protein interaction networks. She then did a postdoctoral stay at the Spanish National Cancer Research Center, where she worked on projects related to cancer genomics. In 2010, she obtained a permanent CNRS researcher position to work in the Marseille Mathematics Institute in the Mathematics for Systems Biology team. And in 2018, she started animating her own research group dedicated to systems biomedicine in the Marseille Medical Genetics Unit. And her interest is to develop computational methods to study human diseases with a particular focus on data integration as well as on rare and comorbid diseases. So thanks a lot, Anaïs, for being here to present your work.
Thank you very much. Everything was true, so thank you. I do have this big bottle of wine. It's a teaser, so you have to listen to me for the whole talk to know why I do have this big bottle of wine. It's a trick. I'm going to talk today about the work we are doing around the development of network-based methods for data integration, in particular in the field of rare genetic diseases. So you all know that we are currently facing a deluge of data. I'm going to talk here about data for biomedicine and biology in general. So it's true, we have a lot of more data in any scientific field, but it's particularly true in biomedicine and in biology. And working or focusing on molecular and cellular data, we have all the omics data, for instance. So we do have transcriptomics, proteomics, genomics, interactomics, and we have, so thanks to the development of methodologies, we have new data all the time. So currently, for instance, we do have spatial transcriptomics and single cell data, for instance. But in fact, we do not have only omics data. We currently have a deluge of data in many other forms. So we do have lab results, for instance. We have images such as MRI. We have video records. We do have a health database, such as a hospital database, but also database such as the French Social Security medical administrative database. We have other type of signals, electrocardiogram, electroencephalograms, for instance, and even more and more data coming from a wearable device, such as phone and watch, for instance. So, and the pace of data production is increasing. So overall, we currently have an accumulation of biomedical data set that is unprecedented, and it's yielding huge opportunity to better understand the diseases, their evolution, to try to find new treatment, and so on and so forth. But together with having access to all these data sets, the data sets are large and complex, and so we do have also huge challenges in the development of methods to try to extract knowledge from these different data sets. And for that, we need proper computational strategies. And this is true for any scientific field, but we do have specific challenges in biomedicine. So I'm not going to talk today about the challenges related to data access, which are really big challenges, but I do not have any solution for that. It's more legal or juridic stuff. But we still have other types of challenges. The first one is it really big data? Because when you have, for instance, 20,000 features, 20,000 genes, but measures in 10 samples, it's not really big data. It's what some people call it fat data, because we have many variables, but very few samples. And in other cases, you can also have a few variables and many samples. So quite often, it's a particular type of large data, but not necessarily big data. So many methods are not really appropriate. We also do face some challenges related to data quality. We often work with human data sets, so we have problems with missing data, limitation, biased data, and this is very important in biomedicine. And also something that Carl mentioned yesterday, we do have some challenges related to explainability and interpretability, because it's not possible, if you have to take some medical decisions, it's not possible to have black boxes. So you really need to have models that can explain themselves, so why they make some predictions. So my point today is to try to convince you somehow, if you're not already convinced, that networks approaches, network-based strategy can really help solving or working on all these challenges in data analysis, interpretation, and so on. So why do I think that networks are a good strategy? It's because networks are great. Okay, so if you have to remember something, it's that. So networks, if I do some definition, networks is a set of nodes and edges. So the nodes could be, for instance, protein, and the edges could be protein-protein interactions. But in fact, it could be other type of objects. The nodes could be patients, and the edges could be patient similarity. Or the nodes could be diseases, and the edges could be phenotypic similarities in between the diseases. So you can build many different networks, and they are central to the understanding of complex systems. You know, complex systems, it's systems where the global behavior cannot be understood from the understanding of each component. You really do need to have the interactions to understand the system. And for instance, if we think about this small network here being a protein-protein interaction network, having only the protein, you would not understand the function. But mapping all the protein-protein interaction, you have a first glance of the functional unit of the biological process of the system. So it's very interesting to try to link genotype and phenotype, for instance. Second point is that networks are intuitive. Because when you draw or you do a doodle of a network, when you have a collaboration with biologists, you still directly understand what you are talking about. So it's very intuitive and very easy to discuss around a network. And also, I think importantly, from the computational point of view, is that networks are very good scaffold for data integration. So for instance, here in this small network, I can imagine that the nodes are genes or proteins. We consider them the same. And you can integrate different type of interactions. You can put some protein-protein interactions, but you can also put co-expression information, pathway relationships, and different types of interactions. So you can integrate heterogeneous data. But you can also integrate network data with quantitative data on the nodes. So for instance, let's say you have a protein-protein interaction network, but the color here represents differential abundance of the proteins in different contexts. So you would be able to integrate in the same system data on quantitative data on the nodes and interaction information. And this is very interesting when you want to integrate like a new data set you just produced, a transcriptomics or proteomics data sets with existing prior knowledge that is often represented as network, such as existing protein interactions, for instance. And overall, the last point is that when you have your data represented as a network, it opens the toolbox of graph theory for the analysis of the network. So you will have access to many different algorithms for the analysis of biological networks, okay? So given all these advantages, the field of network biology is quite diverse and quite large and has had a lot of success. When I was trying to make some categories, what are the network approaches, I came up with maybe 3 main categories. The first category could be the modeling of strategy based on modeling networks. Second category, strategy based on the inference of network, and then a strategy based on the mining of networks. The network modeling strategy, the idea is to explicitly model the knowledge. So based on expert knowledge, on literature, you will explicitly write down what are the components, what are the regulations, and what are the rules. It's rule-based learning. What are the rules that are behind? And so this is really like a bottom-up approach. You start from knowledge and you try to build your model. So it could be, for instance, used for clinical decision support or signaling pathway modeling. In the case of network inference, the strategy here is to build a network from often quantitative data. So you start with measurement, it could be transcriptomics, for instance, and you try to infer the relationships either in between the variable or in between the samples. Here I draw a small undirected network, but it could be, for instance, the inference of a directed network with regulation information. And finally, network mining strategy. This is different because here the network is the input, whereas in network inference, the network is the output of the method. So here the network is the input. You can take a network from large-scale to hybrid screenings that has been constructed from experimental data, for instance. And you will have to develop methods to try to extract the knowledge that is present in these big networks. So now I'm going to try to show you different works we have been doing around these three different strategies for network analysis. So first, focusing on network modeling. So I will not enter into the detail, but this is a very important and successful field in network biology with a strong community in France in particular. So I will just quickly mention some work we are doing around that. So we are interested in developing Boolean models. Boolean models are models in which, so here the nodes correspond to proteins, and the nodes can have two values. They can be either 0 if they are not activated, or 1 if they are activated, okay? And then you will map the regulations. So you will map the nodes, you will map the regulation, and then you will have to model the rules. So for instance, you can say that this node is activated, is at 1, if this node and this node are also activated. So you will write down the formula. It could be also an OR. I will have this node activated if this node or this node are activated. Vous voyez pas ma... I don't know, I have no idea to do. It's I have no idea how to do that. And the thing is that I think we might break something. Ouais, oh, trop fort. Merci. Okay, so I will just say that back. So for instance, you will say you will define all the rules of the model saying like this node is activated, is at 1, if and only if this node and this node are also at 1. Or you can say, depending on your expertise, that you will have this node at 1 if this node or this node are at 1. So you see, you precisely check literature, discuss with experts, and try to explicitly write down all the rules of the model. So we are currently doing that for this particular disease. It's called dyspharinopathies. Sorry, there is a... So dyspharinopathies are diseases, myopathies caused by mutation in the dysferlin protein. And this protein is involved in membrane repair in muscle cells, but its dynamics is not really well known. So discussing with experts and reading papers, we have constructed this model which contains proteins involved in membrane repair, in necrosis, and in regeneration. And we're currently fine-tuning all the rules. And the idea at the end of the day will be to use the model to better understand, to precisely understand the role of the dysferlin in this repair pathway, and how its deregulation simulate its deregulation or its knockout in patients. Okay, so this was a quick example of network modeling. I'm gonna now switch to network inference. As I was saying, network inference, the idea is to build a network out of quantitative data. And the rationale is that once you have your data represented as a network, the analysis is going to be easier because you will have access to the toolbox of graph theory, okay? So as an example of a method based on network inference, I would like to show you the work we have been doing about using medico-administrative databases. So these databases, such as the French Social Security, are databases that are created to monitor what we all get refund for. So for instance, here, it's a sampling from the French Social Security. It's called Échantillon généraliste des bénéficiaires. And you have the data, you have the different patients, you have the date of prescription of a given drug, and what is the drug. And so the data we all have in the French Social Security. So you see, this is not clinical or medical data because we have no code for disease, we have no code for medical code or anything. It's just what you get refund, the drug you get refund for. But still, it's a huge amount of information about the health of the French population because you have all the drugs that we might take. So the question is, how do we extract knowledge from this database? And what we wanted to do is to try to identify clusters of similar patients. Because a rationale was that if you have cluster of patients that have the same age and that take similar drug, it might be a proxy for patients sharing the same disease. But this is not easy because the data, they are temporal data, heterogeneous missing data, and so on. So what we propose is to do that with a network approach. So we propose to build networks of patients. So for instance, here we focus on patients age 60 years old. And our idea is to build a network in which, so here each node is a patient and the edges are, if they are very similar. And we compute the similarity with a cosine similarity using the data I presented before. So we have the different patients. So here it's only patient age 60. And we have the different drugs, and we have the number of drug boxes for which they get refund for when they were 60 years old. So it's just a cosine similarity in between these different vectors. And then we put a threshold and we obtain this network. So what is cool with having the data represented as network is first that you do not use any more raw data. And for privacy reasons, that could be interesting because now it's only relative data. And also, you can see that represented as a network, you find or you see some communities, some clusters. So it seems that the network representation is highlighting some signal or information. And now it's represented as a network, we can use algorithm to try to analyze it. And the easiest algorithm here is community identification algorithm. And in fact, if you identify communities in this network, it gives all these colors correspond to different communities. Okay, so group of patients that are more similar together than with the other patients. This is for patients age 60 years old, but we have longitudinal data. So what we did is construct different network layers for the different ages. So we have the network at 60 years old, the network at 61, and so on and so forth. And in each network, we identify the clusters, and then we just have to track the clusters over ages, and this allows identifying patient trajectories. So here, for instance, are 3 different patient trajectories. So we have the different ages and the cluster that are tracked over time. So this trajectory, for instance, is patient taking this drug, which is warfarin, and which is a drug that was used for people having blood clots, and they would take that all the time after having a blood clot. Other trajectory, for instance, this one, it's interesting because it's patient taking 2 types of drugs. They are taking aspirin and clopidogrel, and the following year, they take only 1 drug. And in fact, it's what people have when they have a heart attack. For 1 year, they have 2 drugs, and they switch to only 1. So it seemed that we didn't use medical data here. We use only what people get refund for, and we are still able to identify clusters of patients and trajectory that makes sense from a clinical point of view. So it's a way of, taking advantage of what is in the social security database. And what we want to do next with that is link this patient cluster with genetic background information that we might have for some of the patients. Okay, so I presented already the first 2 network strategy. I will now switch to network mining, which is in fact the majority of the approaches we are developing in the team. So here, the starting point is we already have very big networks, and we want to extract information from these very big networks. So for the next few slides, I'm going to talk about mainly molecular networks, so networks in which the nodes are genes or proteins, which I consider the same here. Historically, these networks are often called interactome, and it's protein-protein interaction networks. So at the very beginning, they were quite criticized, but the technology evolved a lot. Now they are much more reliable. And what is interesting is that in these big networks, you do have a lot of data, a lot of information about protein cellular functioning, their biological process, and so on. And even for proteins that have few annotations that are not very well known. So it's a very interesting data set, but you see it's quite a nightmare. It's very complex, and you don't really know how to see biological processes from these data sets. So there comes computational biology and different algorithms really dedicated to extract knowledge from these big data sets. And among the different strategies, I think one is quite famous and widely used is the strategy of identifying communities from the large-scale interactome networks. So communities, it's kind of the functional unit of the network. So it's subgroups, subnetworks, group of proteins that share a lot of interaction that might be involved in same pathway, processes, and this kind of thing. But you have different ways of identifying communities from network. One way is to to partition completely the full network. So you try to cut and split all the nodes in different clusters, in different communities. One algorithm to do that is now very famous because it's widely used in single-cell data analysis. It's the Louvain algorithm or Leiden algorithm, and it's exactly the idea. You try to partition a network. But there are other methods, and another set of algorithms I really like are algorithms that are dedicated to try to extract communities around nodes of interest. So here you focus on one node you are really interested in. It could be your favorite gene, it could be a gene mutated in a disease, and you want to study that. So you already start from a seed node of interest. For both these two types of community identification, the only input is the network. So it's using only information from the topology of the network. But there are other sets of algorithms, which I call here active module, in which you will integrate your network data with other quantitative information on the nodes. Could be transcriptomics, could be proteomics, could be GWAS p-value, for instance. And you try to find, it's sometimes called hotspots, so zones of the network of interest because they contain a lot of genes differentially expressed, protein differentially abundant, and this kind of thing. And these are methods dedicated, so-called active module identification methods. Today, I'm not going to talk about network partitioning, but I'm going to present two algorithms here to identify communities around node of interest and to identify active modules. So first, communities around node of interest. One very fancy algorithm to do that is called a random walk with restart. So it's an algorithm that is widely used in bioinformatics to explore network around seeds. And I will try to show you how it works. The way the algorithm is working is that it starts from a seed node. So as I was saying, your gene of interest. And it simulates a walker randomly walking into the network. So if I'm here, I can go there, there, and there. Let's say I'm here, I can go there, 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 or there. And at each step, my walker is randomly walking into the network. But at each steps, it also has a non-zero probability to jump back to the seed. So it means that I'm going to walk like that, and sometimes I will jump back to the seed. And you see that if I do that a lot of time, there's some nodes that are close to the seed, that are highly connected. They might be visited more by the worker than other nodes that are far away from the seed and less visited. And this is a very smart way of deriving a kind of distance score on the network with respect to the seed. So here, for instance, in this small toy example, I have scores for these different nodes with respect to this seed. And you see that it's different from just computing the number of edges, because this node and this node, they are distance 1 to the seed. But this one has a higher score because, in fact, it's more connected. So here it's just a toy example. Imagine your seed is hidden in this very big network. It's a way to give score to all the nodes of the network with respect to the seed, even in this big crowded network. And you can have more than one seed, so it's very useful. So it has been used in many different contexts in bioinformatics, because once you have the score, you can do node prioritization, you can do ranking, and you can do a lot of subsequent analysis, such as community identification, network embedding, and many other strategies. So it has been used to study cellular function, to study gene disease association, for instance. But it has mainly been applied to protein-protein interaction networks. And what we have been doing is try to consider more than one network, because in fact, we do not have access only to protein-protein interaction network. We can build network from molecular complexes that are retrieved from immunoprecipitation experiments. You can also build a network from pathway data from curated databases. And you can also, as I was mentioning previously, you can also infer networks from different types of omics data. Let's say you have here the nodes are genes, and the edges are correlation of expression taken from or computed from RNA-seq experiment, for instance. So what we have been doing is extend the random walk algorithm so it can navigate multiplex networks. So networks in which the nodes are the same, but the edges are different. So here it's always the same nodes, it's genes and proteins, but I have different type of edges, co-expression, protein-protein interaction, and pathway, for instance. So now the walker can navigate in one layer, but it can also jump from one layer to another because it's the same nodes. It's called the replica nodes. So it means that now we can derive a score, a random walk with restart score that can be used in many different applications, but using multiplex networks as input. So you can use that for community identification, embedding, and so on. What we have been showing is that using different interaction sources was increasing the amount of information we were able to extract from the network to predict function, to predict gene disease association, and for different bioinformatics tasks. So it's really worth trying to consider different interaction sources when you develop some network-based methods, let's say. I will show you 2 use cases on application in which we have been using this network to try to study some genetic diseases. The first one is muscular dystrophies. It's a use case based on muscular dystrophies. So muscular dystrophies are pathology of the skeletal muscle, and what is striking is that depending on the mutated genes, you can have different onsets. So you do have some mutated genes that give a distal onset. So the first muscle affected are the muscles of the extremities of the limbs. And you have other genes that when mutated, the disease has a proximal onset. So in this case, it's skeletal muscle of the center of the body that are affected. And taking a gene panel that is used for diagnosis, we identify 11 genes that when mutated give a distal onset, and 19 genes that when mutated give a proximal onset. And it's always skeletal muscles, so it's very hard to understand what are the differences in between these 2 sets of genes. So we decided to do this random walk strategy using these 2 different sets of genes as seeds to recover their communities. So using 11 distal genes in the random walk using 3 biological networks, we recover the distal community. And using the 19 proximal genes in the random walk, we recovered the proximal community. These 2 communities do not overlap. It's not the same subnetworks, it's different subnetworks. So it means that the different set of genes might be associated with different biological processes, different functions, okay? So to try to dig a bit into that, we did some enrichment analysis of the 2 different communities. And in fact, we recovered, so for using, for instance, subcellular localization annotation in gene ontology, that the community of distal gene is enriched in gene coding proteins located in the sarcomere, whereas the community of proximal genes is enriched in gene coding for protein located in the sarcolemma. So indeed, it seems we still don't understand why there are 2 different onsets, but at least the different set of genes are associated with different biological processes. We still need to make the link between why sarcomere is doing distal and sarcolemma is doing proximal. The biologists don't really know neither, but it's at least there is something at the subcellular level that is interesting. I want to say it's a parenthesis, a few words on this figure, because it's the tool and the figure that helped us define the different, compare the enrichments, and it has been obtained with a tool we developed that is called ORSUM. And to ORSUM, you give your enrichment results. So I gave the enrichment results for distal community and the enrichment result for proximal community. And ORSUM is making a summary and removing the redundancy in the enrichment results and making a comparison in between the 2 enrichment lists. So for instance, here, sarcolemma annotation, gene ontology annotation, it's not significant in the distal community, and it's in the first quartile of significant p-values in the proximal community. So ORSUM is quite convenient when you need to compare enrichment list. End of the parenthesis. For the second use case on random walk to better understand genetic disease, I will advise you to go to discuss with Cécile Bust, who is here. She made this type of methods to study premature aging. Yeah, she's there. to study premature aging diseases. So premature aging diseases are diseases for which you have a premature aging phenotype. So you might have seen pictures of kids with a progeria, for instance. But in fact, there are many different premature aging diseases, and they are caused by mutation in many different genes. So we were wondering, as long as they have the same phenotype, are the different genes involved in the same function, or is it different functions? And to know the answer, you have to go to see Cécile, and she was going to do a flash talk just after. Okay, going back to this slide, I was mentioning here multiplex networks. So it's different types of networks, but the edges are always genes or proteins that we consider the same. It's different edges, but the same nodes. And in fact, we would like to work with other type of data. We would like to integrate and explore more data and more heterogeneous data. So we want to consider networks here. Each bubble represents a network, and we would like to have networks with nodes being genes and network with nodes being diseases or drugs or side effects or a lot of type of available biological information. And networks are quite convenient because you can connect these different type of information with what is called bipartite interactions, so interactions between different types of nodes. And in fact, we want to explore beyond multiplex network. We want to explore multi-layer network, which are also known here as knowledge graph. And we want to have methods to extract information from this. It's more complex, but it's richer interaction data sets. So we have been working on extending the random walk with restart so it can navigate this more complex multi-layer network. It was not easy because you have a lot of matrix, a lot of combination of matrices, a lot of parameters, but we have a tool that is working. It's called MultiXrank, and it's available on the GitHub to explore, to do random walk around seeds on these more complex networks. And so here too, you can use the random walk score for many different applications. It's really, it can be used for community identification, embedding, and so on. It's really what is nice with random walk is that you can really do anything with the vectors of scores after. And if you want to have example of that, this time you can go to discuss with Galadriel Brière, who is here. And in fact, she has been applying MultiXrank in many different use cases and contexts to do node prioritization, also to do gene disease prediction with supervised learning, and also to compare the different gene profile from different diseases. So if you want to see different usage of the random walk with restart, it's a very good idea to go discussing with her. I'm going back to this slide. So I said I will not discuss partitioning. I present you my favorite random walk with restart algorithm to study communities around node of interest. And now I would like to say a few words on active modules. So here we do not use only the network topology, but we try to integrate network topology with quantitative information on the nodes. And the idea is that you would try to integrate a biological networks with, let's say, for instance, RNA-seq transcriptomics data, where I have a case control with differential expression in between patient and control. And I would like to combine both type of information to find subnetworks of interest, like our active modules. That could be something like that. It's a zone of the network that is enriched in interaction and that is enriched in nodes differentially expressed. Another way to see the same question is to say, I want to do a transcriptomics analysis, but I want to enhance the signal by using network prior knowledge. Okay, so it's a way of using what is already known in the literature, the network, to better analyze your transcriptomics data sets. Finding active modules is not easy. It's an NP-complete problem, and there have been different algorithms. However, the methods, at least to our knowledge, they did not consider the density of interaction inside the community. They were using only one protein-protein interaction network, and you might have guessed that I like using more than one network. And the method were not tested on the RNA-seq data. So we propose here a new algorithm, which is a multi-objective genetic algorithm to try to identify active modules from multiplex networks. So I don't know if you know about genetic algorithm. It's quite fancy algorithm. I will guide you through their process. So in this algorithm, we start with an initial population, a random population of solutions. Our solutions here are subnetworks. So we start by fetching subnetworks from the network completely randomly. Let's say we have 1,000 potential subnetworks. We then rank this subnetwork, we rank the population based on our two objectives that I will describe in the next slide. Once you have the ranking, you choose the parents. You try to select two parents, which are among the best solutions according to your ranking. And from the two parents, you create two children. Let's say one parent here, one parent here. I just cut and cross over the parents to have two children that are represented here. So you can also here introduce some mutation, adding a node or removing a node, for instance. So you now have a population with 1,000 parents and 1,000 children that are different from the parents. You rank again this population using the same criteria, the same two objectives I'm going to mention, and you select the new population, taking the half better solutions. Okay. And you go back, choosing two parents, and small pieces by small pieces, you are evolving new solutions, exploring the space. And you stop when you reach stopping criteria, like a number of generation, for instance. So two steps are based on ranking the population. It's very important steps. So how do we do that? We have two objectives we want to maximize, because we want to maximize the score of the node, which is the differential expression in the transcriptomics analysis. We want to have a maximum of differential expression and significant differential expression. And we also want to maximize the density of interactions. So we want to have community that contain a lot of interactions coming from the multiplex network. So we want to maximize these two objectives at the same time. And so we rank the solutions, we rank the subnetworks according to these two objectives. And as we have two objectives, we use Pareto dominance to rank the solutions. So here you have a plot in which you map the differential expression, so the average node score, and the density of interaction. Each dot is a solution, it's a subnetwork. And you will rank your solution based on the Pareto dominance. So the first Pareto front will contain all the solutions that are non-dominated. So it means that there is no other solution that is better for the two different scores. So for instance, this solution, there is no other solution that will be better for the differential expression and the density. And you use that to select and make your population evolve toward better solutions. And so at the end of the day, your final set of solutions is the final first Pareto front. So for instance, here we have a final solution, which is a trade-off in between density and differential expression. But you could also be interested in this solution, which is better on differential expression, but not so good on density, or this solution, which is the other way around. Okay? So it's a very interesting algorithm to try to explore the search space. It's quite long. This is the main point because it takes time to evolve, but it's working quite well. And we have been using that, for instance, to study another myopathy, which is called facioscapulohumeral dystrophy, so FSHD. This myopathy is not caused by mutation in a gene, but it's a hypomethylation of a genomic region, which is called D4Z4. And the consequences of this hypomethylation are poorly described. So we had an RNA-seq data set in muscle fibers derived from induced pluripotent stem cells in patient and control. And when you do the differential RNA-seq analysis directly, there were only a few differentially expressed genes, like 200, and no enrichment in pathways. So we decided to apply our genetic algorithm, which, by the way, is called Mogamoon, to combine this RNA-seq data with molecular network in Mogamoon, and we identified about 14 different active modules, such as the one presented here, where you see you have a lot of interactions in between genes that are downregulated, and in fact, it's a module of genes involved in calcium uptake. So I don't know if you can see. Oh, the video is not working. We spent so much time testing if the video was working. It's a pity, but well, so it's yeah. Stylet, surligneur, machin. No, but otherwise it's not. I don't know where to click. No, but it's not a big deal. Because then you will be disappointed. It's just some cells showing some green lights. So. Ah non, c'était avant. Voilà. Ah si, ça marche. Okay. So we found that we might have a downregulation of processes related to calcium uptake in muscle cells, and this has been validated experimentally with calcium measurement on muscle cells. Okay. So you've seen that we have different strategies for analyzing networks, some strategy based on topology alone, some strategy combining topology and other type of information. In fact, choosing a strategy is really dependent on your question and on the data you have. And it's something in between these two strategies, a random walk and active module. If you want to think about that and to discuss about that, you can go discuss with Morgane Teresol, because she has a poster where she implemented the two different strategies. It's not finished, there's a fourth one, where she implemented the two strategies to try to study the relationships in between genes that are involved in rare disease pathways and genes that are targets of chemicals. So with the active module and the random walk strategies, so you can have a comparison of the two approaches on the same question. So I hope I convinced you that networks are great. I try to show different examples on how we can use them for data integration of heterogeneous edges, of heterogeneous node and edges, to use them as previous knowledge, and to also show you different algorithms that are part of the toolbox of graph theory that can really help extracting more information from data. And it might not be clear, but we do not only work on network in the team, in fact. So we also have different approaches. So we are trying to do systems biomedicine. And apart from networks, we also have some work on matrix factorization and autoencoders, where we work with matrices and not networks. But in any case, we are trying to, the main goal is to try to extract information from biomedical data. I've been showing here RNA-seq and intractome data. But we are working more and more with spatial transcriptomics and single cell data. And I will advertise for the last post there of Céline Chevalier, where she analyzed a very interesting data set of single cell with a time series single cell data obtained in gastruloid for health development. And this raised very specific bioinformatics challenges. So you can discuss these challenges with her. And finally, in the application, so you have noticed that I discussed data for public health from the French Social Security, but that all the other approaches I mentioned are on rare genetic diseases, because I think this is a very important point. Rare genetic diseases, they are very rare, but together there are more than 6,000 different diseases. So it's a huge number of patients. It's a huge challenge. And I think there are not so much bioinformatician, computational biology working on this field. And I think it's particularly interesting for us, because all the data production, data integration, it's raising challenges, but it's gonna bring solutions in biomedicine in general. But I really think we do have to invest also the field of rare diseases, because I don't know, that many patients, we do not even have a diagnosis. We don't know the mutated genes for half of the patients. They have a huge heterogeneity we don't really understand, such as distal and proximal phenotypes. We don't get the pathophysiological mechanisms, and we don't have treatments. So I think there is a huge opportunities to better understand these diseases, and it's what we will do in the team for the upcoming years. So I have to thank all my team. Many of them are here, so please go discuss with them. They are really expert on their different topics. Yeah, and that's it. And I have to explain you why I have this big bottle of wine. So in fact, it's the bottle of Baudot I won as for poster prize in 2006 in JOBIM in Bordeaux. And it was lost for 17 years. It was lost. Because, you know, life happens. So I was pregnant, I moved for a postdoc, and I found this bottle of wine in my mother's basement the day before I received the invitation for a keynote here in JOBIM. And I thought it was a really good clin d'œil, because it's nice to be part of a community for such a long time. And so the idea that for people in Nice, we will drink this bottle of wine tonight. Thank you very much.
Yes, thanks very much, Anaïs, for the great talk. So for the question, should I, there are plenty of questions, okay. So should I read the question? So thank you for this very interesting talk. In the SNDS network analysis, I did not understand how you make the link between the clusters from one age n to age n+1. Is it based on the number of shared patients between the two clusters? How did you determine the threshold? And did you observe sometimes a repartition of one cluster at age n into two clusters in the next network age n+1?
So yeah, exactly. The tracking of cluster from n to n+1 is based on the number of patients, considering that we have some patients that get out of the cohort because they are not in the same time frame and so on, but it's based on the number of patients, and then we track the majority. So sometimes we have a split in two, but this did not happen a lot. In fact, usually it's like 60–70% going somewhere, and the other either part are dead, part are out of the cohort, and so on. So this answers, I think, the first and the last question, but I might miss the one in the middle.
In the middle, how did you determine the threshold?
So the threshold, so if the idea of having a threshold for the cosine similarity, it was based on 0.8 cosine similarity to build the network. But if the threshold is for cluster tracking, it was a majority rule, where the majority of the patients were going, but usually it was more than 50%.
Okay. From the sample, so in multiplex networks, how do you deal with the different compositions in nodes of each level? Do you reduce the multiplex to the shared nodes between all layers? Do you complete with added links between layers?
So no, we do not reduce to the intersection, because the intersection between 4 layers is like 20 edges. So this is not possible. In fact, in the definition of the multiplex network, you say that all the layers contain the same nodes. So you have exactly the same node in all the layers, which is the nodes in the union of the different networks. But in some layers, they have no interaction. So they are isolated nodes. But the full set of nodes is the union of all network nodes.
Okay. So from someone else, thanks for the great talk. You mentioned some work where you work more on matrices rather than networks. Could you explain the differences you make between, sorry, the last part of the question is missing. Could you explain the differences you make between these two representations? Sorry if I misunderstood.
No, it's okay. In fact, a matrix and a network is two sides of the same coin. It's just a different way of representing. But then you have different tools. So when you build a network out of data, you have to make some thresholding and some choices, except if you have protein-protein interaction. So you could work with adjacency matrices, for instance, and some tools can work a bit the same. But all the graph theory methods, such as random walk and so on, they work more on graph objects. And other methods, such as matrix factorization, they work more on matrix representation. Of course, you could do a matrix factorization with an adjacency matrix, but it's not going to be really useful, and it's not going to bring added value. But at the end of the day, it's similar sets of strategy, let's say.
Okay, thank you. And there are questions. There is okay. There are questions here.
Yeah, thanks. Very nice talk. So I'm very happy to see networks used like this. I mean, I like working with them a lot as well. But so my question is, so for example, you were showing these small network communities that you could extract, that you actually validated. But so in general, once you have this type of solution network, for me, this is where, okay, what do we do now? What do you usually do, and how do you use those network solutions?
Yeah, that's very interesting, because we do not have any gold standard. We do not know the true communities, the true biological processes. So we have to have proxies. So either we validate them with biologists, or either we have proxies. So what I usually do is try to check for the enrichment in the overlap with gene ontology, biological processes. It's what is usually done. But I agree that it's sometimes a bit criticized, because finally you could shuffle a bit and obtain also significant results. So it's hard to compare. For me, I think it's not a final set of biological process. It's a way to analyze the network and to provide some more interpretable subnetworks instead of having the big nodes. But we cannot say that it is true. And in addition, what is important is that all the biological networks I showed, the molecular one, they do not have spatial and temporal context. It's relationships. It's a relationship object. But finally, if two nodes are never expressed in the same place at the same time, it's not going to happen. So I think what will be the next steps is try to integrate more this type of data with dynamics and spatial information.
Any other question here? No? Maybe at other sites? Are there questions in the other sites?
So you are nice, because last time I presented something similar, and the first question I had, some guy saying like, I don't think networks are great. So.
I had some questions. So I was wondering whether you were planning or you already tried to incorporate regulatory information in your networks.
So that's a long-term discussion, because I'm working very closely with Elisabeth Remy, and she's coming from the modeling side. So she's really doing all this bottom-up approach, and I'm going more from the mining side. And so we are trying to converge for many years. So in some inference, you can do some regulatory network. And in fact, the random walk we developed can work with directed signed networks. It's just that we don't use it so far, but it's something I will really like to do, in particular working on single cell data.
Okay, thank you. I was also wondering if when you describe the identification of clusters of patients with the cosine similarity, if you try to use other measures of similarity?
Yeah, we tried 4 or 5. The cosine was the one giving very different, more visual clusters, but we tried with different distances. And in fact, we have a very new paper on bioRxiv for 2 weeks, in which we try to, so here it was only cosine similarity for the number of boxes of drug you get refund for. But we tried with a new method in which, in addition to computing the number of boxes, we also used the ATC classification, which is the drug classification. So the idea being like, if 2 patients take a similar drug, we want them to be closer than 2 patients taking different drugs. So in addition, with the number of boxes, we consider the relationship, so the relationship in between the different boxes. Okay. And this improves the classification. So it's a way of using more prior knowledge in the data analysis is increasing
okay. Oh, nice.
the information.
Okay.
Yeah, brand new on bioRxiv.
Very nice. Any other question? Okay. There is one. 2 others. Okay.
Just a moment, so I'm interested in Orsum. So you were saying that it was helping with the redundancy of the enrichment result, and I didn't fully understand. So what type of redundancy?
So gene ontology, when you do a gene ontology enrichment, you will have many redundant annotation, like a transcription if the weather is good, transcription if the weather is bad. And so the way Ozan implemented that is to check finally the more precise annotation with the best p-value and removing the others. But if you do that for 2 different enrichment results, it do that for both lists. So it will end up with 1 annotation for the 2 lists, so you can compare the different lists. So it's working. We use it a lot because it's working quite easily, in fact.
There is another one.
Eric, right?
Thanks for the talk first. In genetic algorithm, you have some aspects that you can design or choose. This is the type of operation you can do on your network. Here, you have shown some slide. So what sort of mix or recombination you can do between subnetworks, and how do you choose those? So the type of operation that you allow in your genetic algorithm, and why?
Yeah, so that was a huge part of designing the algorithm, because the existing, so it's based on NSG, it's based on an existing genetic algorithm, NSGA2, but which was not designed for graphs. So we had to implement a graph version. So what we did is that we said the parents need to have at least 1 node in common, so we can recombine them. So this one was one of the strong adaptation.
The color code.
Yeah, it's the color code. Yeah,
Okay,
exactly.
okay.
And then we said that we put some parameters. We can add one node, we can remove one node. So this is something we had to decide. But it's converging. It's converging. The thing is that it's so the fact that the parents need to have one node in common is really important. Otherwise, we cannot do the crossover. So this was the stronger parameter. But then changing one mutation, two mutation, it was not changing a lot. The only thing is that here it's very long. It's taking 30 hours to converge. And so this, we don't really know. I don't know. Maybe Laurent has new ideas. Laurent?
That's a good idea. Okay. And why is it so long?
Ask the computer scientist. Yeah, the exploration space is quite huge because the network has about 10,000 nodes. But I think it's because you have generation and generation and generation. I don't know, Laurent, if you have a better answer to that. Why it's so long?
To do a crossover, we have to be sure that the network share one edge. So our neighbor of each other. So that there are many tests of neighborhood and intersection of potential networks. And so this kind of lookup should be improved. Okay.
The code is on GitHub, so anyone having an idea to try to improve the time?
So I was wondering whether you can add weights to your edges to help your worker to drive around the network using, preliminary data or knowledges or other stuff,
So the new version, Multix-Rank, can work with weighted networks. Then the thing is that for this work, we already had to prove that considering different network sources was better. And we didn't want to include weight in addition, because then you have to put what is the influence of the weight and so on. So either you could use weighted networks, but you can also weight the random walk. For instance, you have 4 layers and you do not believe in the co-expression. You think it's interesting, but maybe it's more noisy. And so you can give more restart weight. You can say that it will never, or it will not restart a lot in this layer. So you can also weight your source like that. This could be done.
Yeah, there was a question here, and I think maybe the last one.
Yeah, I was just curious. So how does this one compare to HotNet, for example?
So I will have to. So the thing is that HotNet, it's a simulated annealing, right? So if I remember correctly, we compared it in the paper. So we compared with 2 existing algorithms, and one was giving huge subnetworks, like containing 1,000 nodes, and the other one was giving very small subnetworks. I don't remember which one is doing that. The thing is that HotNet is, I think the initial paper is 2002, and it was not tested on RNA-seq data, and in this setting, it was not working very well. But the idea is it's the same strategy, it's active module identification. But now I discussed with Baudot from Pasteur, Baudot Shikovsky. He was one of the developers of HotNet, and now they have a new method that they call Lean, which is identifying subnetwork for connectivity. And there's another method which is called Domino. Morgan has this in her poster.
Okay, thanks very much, Anaïs, for a great talk.
So I wait for you tonight for the wine. I hope it will not be terrible.